I want to actually continue the conversation and do a little bit of a deeper dive from a regulatory and quality consideration perspective. Um, and so perhaps Julie, we can continue the conversation. What are, some of those regulatory hurdles that we're talking about that are unique to iPSC-based therapies, particularly around donor cell sourcing, genetic modification, long-term safety, what have you? You know, what are some of those hurdles, and then how can the industry actually work with the regulatory bodies to address these issues? Are there any best practices that our audience might benefit from?
Yeah, you're hitting on a lot of the top high points, which would be, the donor cell sourcing. Informed consent is really quite critical to make sure that you're getting the appropriate information in there. The variability of the donors, the characteristics. Is it important to be able to have a male and a female? Is it important to have different blood types? These might be things that you'd also be looking at. Um, as far as the regulators, they're going to be looking at potential off-target effects, unintended alterations that could impact the cell behavior or patient safety. So I think that's very important. Long-term safety and efficacy. So tumorigenicity is critical. Monitoring, post-market safety, or post-monitoring once they're infused, even in early phase clinical trials. So just how do we navigate this? I think it's early dialogue, but it's also, you have these communities where people are helping to have these discussions with the FDA. So at the ICT, we have a global regulatory team. So if you're part of that membership, you're going to be able to understand what's going on and how you can interact. And also, you might have the opportunity to speak to them one-on-one. Attending these meetings, I had a great opportunity 10 years back when Peter Marks just came into the field, and I met him, and we're inviting FDA to speak at meetings and kick off things to be able to help us understand what they needed. So I think it's really a lot of networking with these organizations and understanding. There's also Alliance for Regenerative Medicine that focuses on a lot of policy as well as FDA, and they usually bring them in. So I think that is really important to have that proactive engagement and early dialogue. Also, clear documentation and transparency. There's a document with the FDA, and there's a lot of guidances that shows you exactly what goes in there. You don't want to waste time putting something in, and they have to keep sending it back to you because you didn't do section 4 or something the way that they wanted to see it. So it is really critical to be able to do that. And, if you have the opportunity, you could also bring in a consultant to be able to help you navigate that process. But I think, those are the important pieces to really be engaged and look at the safety studies, development of biomarkers and things that are going to provide more information on your safety and efficacy of your products.
Sure. All excellent, excellent information. And Erin, I'd like to offer you the opportunity to, I'm sure you echo the sentiments, a lot of what Julie said, but of course, from your perspective, what are some of those unique hurdles that are specific to iPSC-based therapies? And what can our audience do perhaps to work with regulatory bodies to address them?
Yeah, I mean, I think as Julie mentioned, engaging with regulatory authorities early on your specific cell line that you're using, right? There's a lot of guidance documents out there, so educate yourself on the latest guidance. Be part of industry working groups with Alliance for Regenerative Medicine or Bioforum to really understand what the current status is, and then engage with them on your particular cell line, right? It's you have to know your product, and so you have to be able to engage with them on those specific attributes of your starting material, your starting line. I think one of the other unique challenges to iPS-based therapies is whenever you're making your drug product, your final cell, therapeutic cell, is ensuring that you don't have residual pluripotent stem cells in there because of the risk of potential tumorigenicity from those. And so being able to look at your final product in terms of safety, quality, and identity is super important. And I think that the residual PSC potential is one that is particularly unique to this iPS-based field.
Can I just
Same.
echo that with
Of course.
Erin? Because I think, a lot of people question how do we handle that? How, are we selecting? What is the opportunity to handle that contamination, if you will, of different cells? So I think that that's a hot topic, and there's a lot of space for us to be able to improve in that as well.
Sure, absolutely. I want to take this opportunity to remind our audience to please feel free to submit your questions for Erin and Julie. We will be getting to them in just a few minutes here. So if you have questions, submit them via the Q&A tool, and we will try to get to them. One more topic, well, excuse me, one more question under the regulatory and quality considerations umbrella. And Julie, perhaps I'll start with you. We always talk about from a U.S. perspective and working with the FDA, how important is it for industry to align globally on iPSC standards, and what efforts do you see, and if any, emerging to drive harmonization across regions like the U.S., EU, and Asia?
Thanks, Erin. It gives us an opportunity to enhance patient safety and efficacy. They're going to learn something, whether it happens in the EU, Asia, or the US, and why we can't bring that data all together. It's just, I know that we've been trying to drive harmonization, and we do have some of that through our ICH standards, pharmaceutical for human use, and the World Health Organization, establishing international guidelines, but really being able to, partner with someone, say in the EU, and we have synergies where we want to execute on a clinical trial at both sites. Well, it's really looking at harmonizing the regulations, which aren't, and it might be that if you're doing it in the EU, you have to go into full compliance of GMP, and in the US that might be a little bit different. And so it would really help us to harmonize, and if we could have similar standards to be able to execute on it, I think, as well as the industry, consortium that we discussed already, the different organizations, and being able to work with, possibly a set of regulations that are the same. I think it would have an opportunity to really accelerate what we're doing, because there are opportunities for us to execute in different areas and treat different patients. Obviously, when you're in a different country, you're going to have a different genetic profile for patients. So it would be, increasing patient safety, efficacy. It would be an efficient resource use, and to foster innovation that we see in these, in these different areas.
Sure.
Yeah, I agree with what Julie said. I think it's in some cases, it's just a matter of time, right? As we get more and more clinical data, there'll be a greater level of comfort with, okay, this is okay and this is not. But right now, I think that there's apprehension and there's a lot of unknowns. And so until we get more data, that's why it's been a little slow going. But the industry efforts to push for that harmonization, I think, will continue, and I think we'll get there and make strides. But I think it will require a bit more data to help inform how we can come together on something that satisfies everyone's concerns.
Great, thank you. I want to move on to commercialization and access. And so, Erin, perhaps we can start with you. How close would you say are we to realizing off-the-shelf allogeneic iPSC therapies for widespread clinical use? I know we talked about this at the top of our call, but I was hoping we could really get into it here. What milestones do we need to hit? What does the next, what does the roadmap look like?
Yeah, I think, as I mentioned before, I think, progression through the different phases of clinical trials going into phase 3. Obviously, it's great to have some indication of efficacy or clinical proof of concept, and then being able to have the first wave of therapies hit BLA and be able to commercialize it. That's a key milestone. Another milestone that's critically important is to have the conversation with payers about reimbursement. I think in order for these therapies to be adopted, we have to work with hospitals and with payer systems to understand how does this fit into the armamentarium of therapeutic approaches in different areas. I think sometimes that could be disruptive to normal revenue streams for hospitals with the current standard of care. And so we really need to work with the payer system in order to get them to see the potential therapeutic value of these therapies and how over time it can help reduce the burden on the healthcare system by providing these transformative therapies. Again, I think more data will help with this, and I think that those are the types of conversations we really need to help with wider spread clinical use of these therapies.
Certainly, thank you. And Julie, same question for you. I know, as you know, right at the top of the call, you brought this topic up. How close are we to actually realizing off-the-shelf allogeneic iPSC therapies? What's your take?
Yeah, so I mean, I would echo a lot what Erin has said already. I do think that she discussed some of the challenges with reimbursement and so forth. We already discussed manufacturing advancements. I think the more scalable, that's going to help us to accelerate these to the clinic, and the regulatory engagement that we discussed. But key milestones, and as Erin said, phase 3, looking at long-term safety data and efficacy, because we've just seen some of these trials that are starting, especially with the immune cloaked cells. And so the immune compatibility, that definitely is, needs a runway yet. We're just kind of getting into that space. I think cost effectiveness is key as we bring in these opportunities that we've talked about to be able to reduce the cost, and then the market access and reimbursement, which Erin discussed, is critical. And I think a lot of people aren't thinking about it, especially in an academic situation. But we do see with some of these applications how it's not cost effective to bring them in, but we want to be able to treat those patients. So how do we get to that point? There needs to be a larger focus on reimbursement. And then I think education, I'll just say education, because the physicians need to be able to accept it, and the public, they need to be able to understand it. And if they don't understand what an iPSC is, because it's quite complicated, that I think that's going to be a consideration. So I think all of these things would help. It's hard to put a timeline on that. I think we're in early stages, but there's just so much potential to be able to move it forward. So exciting potential. Love to see something move to late stage clinical trials within the next 5 years, and it seems like there would be opportunity for that. But it's really hard to put a exact timeline on that.
Of course, sure. Before we formally move on to our audience Q&A, I do have kind of one final sort of future vision question for both of you. Erin, what emerging technologies or perhaps scientific breakthroughs excite you the most when it comes to unlocking the actual full regenerative potential of iPSCs?
Yeah, I think the continuing marriage of gene editing approaches with iPS technology is just super exciting. I think if you can engineer cells to provide more specificity in targeting or enhance persistence or expression of therapeutic factors, I think you can really leverage a lot of the advantages of iPS technology that we've already talked about with more precise control over what you want those cells to do. I think that there's so many different applications for gene editing technology. I think that is the versatility of that is just incredibly exciting to me, and I think the sky's the limit as far as what you can do and the different applications to partner with those.
Great, thank you. And Julie, for you, if we look ahead a certain number of years, what do you think that or sincerely hope, the iPSC therapy landscape looks like, and what role do you see the Mayo Clinic perhaps playing in shaping that future?
Yeah, no, I think the iPSC field in general is poised to change significantly advancements in how we execute on these biotherapeutics. And hopefully this will come through with, more widespread clinical adoption, enhanced patient applications. We talked about some of these and what are the most common and low-hanging fruit that we might see. Improved safety profiles would be incredible with the cells as we learn more about them. And as Erin described, the gene editing, I mean, that is something, will we see this out 10 years commercialized? I don't know about that, but to be able to see that in clinical trials would be amazing. I actually was at the Diabetes Research Institute for many years in the University of Miami and was at the bedside with a lot of that because we would take the products over to the hospital and would see the patients, and then the patients would actually come and visit us because the patients that got those islets that completely changed their life because they weren't leaving their house or anything, but they had to continue on the immune suppression, which there's a lot of after effects. So to be able to see that and the difference we could make in someone's life, but take away those drugs, that would be incredible. That's really what I hope for the Mayo Clinic. We're starting to look at several different applications. Diabetes is one of those, and neurological diseases as well. It's in very early stages of preclinical development, but we're hoping that we can help with those patients as well. So it's an exciting field with where we are, which gives us this opportunity to hopefully give patients a quality of life that they haven't seen before. But as Erin mentioned, how do we get that cost down? How do we think about reimbursement? So that's a big piece.